Hello everyone. Through this video, we'll be studying the topic Basic Mechanism of Muscle Contraction. The contents of the lecture include sliding filament theory, the cross bridge formation, role of ATP, role of calcium ions, chemical events during contraction, then muscle relaxation and oxygen depth and Cori cycle. What is muscle contraction? Muscle contraction is a complex physiological process which is very much essential for the movement of the body and also for the body to perform vital functions. This happens only through coordinated interactions between the muscle proteins. The most important ones are the actin and the myosin. And here various theories, they explain this mechanism of contraction. And the most important one is sliding filament theory, which has been widely accepted. Sliding filament theory. This is the most widely accepted explanation for the contraction of muscle fibers. According to this theory, the contraction mainly occurs due to the sliding of the thin filaments, which are the actin filaments, over the thick filaments which are present within the muscle fiber. Importantly, the filaments themselves, they do not change their length. Instead, the degree of overlapping between the actin and the myosin, it increases. As compared to this, you can see the degree of overlapping has increased, which leads to the shortening of the sarcomere and then the shortening of the muscle. The basic structural unit involved in the process of muscle contraction according to the sliding filament theory is the sarcomere. So the sarcomere, we know that it lies between two Z lines. During contraction, the sarcomere, it contracts. That is, the Z lines, they are pulled very close together. This leads to the shortening of the sarcomere. So this is the relaxed one, while this is the contracted one. As a result, what happens is the I band, which exclusively consists of the actin filaments, it becomes narrower. It becomes narrower, and the head zone, the region that again consists of only myosin filaments, it gradually disappears. However, A band, which is the length of the myosin filaments, it remains constant because the thick filaments, they do not change in size. This sliding filament theory, it is highly organized and it is very much efficient, that allows muscle fibers mainly to contract smoothly and then generate force. This coordinated shortening of multiple sarcomeres across the length of the muscle, this is the sarcomere, and such contractions happening across the length of the entire muscle leads to contraction of the muscle. Thus, sliding filament theory, it explains how microscopic interactions between proteins result in microscopic muscle movements. The sliding filament theory, it talks about cross bridge formation. So this is the key step that happens between the actin and the myosin filaments, cross bridge formation. This process begins when the myosin heads, they bind to the active sites on the actin filament. This attachment, once it has taken place, that forms the cross bridging, and here it acts like a tiny hook that is connecting the two filaments. Once formed, The myosin heads, they undergo changes, conformational changes, and this we call it as the power stroke. During this step, what happens is the myosin heads, they bend and pull the actin filament towards the center of the sarcomere. They are pulling it towards the center of the sarcomere. So when this pulling is taking place towards the center, here you see that there is shortening of the sarcomere that is taking place. So once the power stroke is done, the myosin heads again, they detach. So how this all happens? Here you see that it's a cyclic of events that take place in the actin-myosin cross-bridge formation. The ATP, it binds to the myosin head, causing its detachment from actin. Once ATP is bound, it detaches from the actin filaments. ATP hydrolyzes to generate energy, where you get ADP and pi plus energy. This re-energizes the myosin head. The myosin head now interacts with the sites, with the binding sites that are open on the actin filaments. So once this is bound, the power stroke happens, the energy is used for movement. So once this is pulled front, the myosin head detaches and again it is ready to bind to ATP. So once again ATP will bind, then ATP is hydrolyzed, giving rise to ADP and phosphate. Now again this changes the orientation. The myosin head with the high energy, it goes and binds to the myosin binding sites of actin filament. So this is a cycle of events which are taking place. So this continues to happen as long as calcium and ATP are available. As you know, if calcium is there, the actin filament heads, their orientation will change because of the troponin and the tropomyosin. And once these are open, then only the myosin head will go and bind there. Now what is the role of ATP? We all know that ATP is very essential for muscle contraction. The myosin head, this contains the ATPase activity and it hydrolyzes ATP to give rise to ADP and inorganic phosphate, releasing energy. This energy, it's mainly used for conformational changes in the myosin head. Okay, then the active site is opened, which then goes and binds to the actin filament. The power stroke happens wherein the actin filaments are pulled inside. Then detachment of myosin from actin will take place. So each cycle of cross-bridge formation, each cycle, it requires one ATP molecule. One ATP molecule. If you look at the sequence of events which are presented here, you see that once the action potential reaches the nerve endings, it is transferred to the muscle fibers across the motor end plate and here the T tubules. Okay, and they receive the signal, transferring it to the sarcoplasmic reticulum. The sarcoplasmic reticulum, it will release the calcium. So this is the actin filaments which have the troponin and the tropomyosin. So these filaments, they have troponin and tropomyosin. Once calcium binds the troponin, then conformational changes will happen. Then the sites open for myosin head to bind. Already this myosin head, it is charged with the energy that is released upon hydrolysis of ATP. And then the muscle contraction takes place. So for every cycle, one ATP molecule is utilized. Coming to role of calcium. So calcium, it is one of the key regulatory element in muscle contraction. When the nerve impulse, it reaches the muscle fiber across the motor end plate, because of the release of acetylcholine, it generates action potential within the muscle that spreads across the sarcolemma and through the T tubules it reaches. Now here, this particular activity, it triggers the sarcoplasmic reticulum to release calcium ions. These released calcium ions, which are now present in the sarcoplasm, they bind to troponin. This causes transformational changes that moves the tropomyosin. So this particular thing, it exposes the active sites of actin. Then the actin filaments, they'll be binding with the myosin heads. So that is how it will be taking place. And calcium plays very important role in exposing the sites, myosin binding sites on the actin filament. Let us summarize the chemical events during muscle contraction. So acetylcholine, it is a neurotransmitter that reaches the neuromuscular junction or the motor end plate. This generates action potential in the muscle fibers. Then as it reaches through the T tubules to the sarcoplasmic reticulum, calcium ions are released from the sarcoplasmic reticulum. This calcium, it binds to the troponin. This binding, it slides the tropomyosin away, exposing actin binding sites. So now you can see formation of actin myosin complex or actomyosin complex. Hydrolysis of ATP contributes to the contraction of muscle fiber. Now, how does muscle relaxation occurs? This occurs only when stimulation stops. The calcium ions, they are actively pumped at the expense of ATP back into the sarcoplasmic reticulum. Now here, as calcium levels are decreasing, you see that the troponin, it returns to its original shape, allowing also tropomyosin to cover the binding sites of the actin. So troponin and tropomyosin, they'll be covering up the active sites of the actin filament. So this particular thing, if it happens, then the myosin actin cross bridge formation will not take place, and hence the muscles, they return to the relaxation state or resting state. Oxygen depth and Cori cycle. So when a person is doing intense exercise, the oxygen supply will not be sufficient. It becomes insufficient to meet the needs of the muscles, leading to anaerobic respiration. So when anaerobic respiration is taking place in the muscles, that will lead to accumulation of lactic acid. This causes muscle fatigue and cramps. So this condition, this entire condition, we call it as oxygen depth. So during recovery, what happens is, most of the lactic acid that is produced, it is converted back to pyruvic acid, while some of it is oxidized to carbon dioxide and water. So this process, it occurs mainly in the liver, and this is known as Cori cycle. Let us summarize the topics that we have studied. Sliding filament theory, cross-bridge formation, role of ATP, role of calcium ions, chemical events during muscle contraction, then muscle relaxation, followed by oxygen depth and Cori cycle. Recommended books for further study include Guyton and Hall Textbook of Medical Physiology, Essentials of Medical Physiology by K. Sembulingam and Prema Sembulingam, or Human Anatomy and Physiology by Elaine and Mereb et al. Or you can study Review of Medical Physiology by Barrett, Susan, or this is the Indian other one, Textbook of Medical Biochemistry and Physiology by M.N. Chatterjee and Rana Shinde. Thank you.